She will be presenting on conservation of a culturally relevant fish at landscape scales, a case study of Pacific lamprey. Dr. Kellie Carim is an aquatic biologist whose research is focused on using genetic information and tools to inform management of freshwater fish and aquatic systems. She received her BA in biology from Carleton College in Northfield, home state, Minnesota, shout out there. And then her PhD in fish and wildlife biology from the University of Montana in Missoula. She may be best known for her work using environmental DNA methods to understand the presence and distribution of aquatic species on both local and landscape scales. And today she'll be presenting on some of that information. And so without further delay, Kellie, I'll hand it off to you. If you would, you can go ahead and share your screen and we can get going. And I guess just one other thing I will mention for everybody who's joined us today, throughout the presentation, please feel free to put them in the chat, and we'll address those at the end of the talk. We'll also be sharing some links in the chat as well, so feel free to click on those and take a look at that as she's presenting and at the end. And yeah, I guess with that, take it away.
Great. So you guys all just see the regular screen, not my presenter view. Okay, great. Yep. Yeah, thanks for the introduction, Karen. I'm super excited to talk to all of you today. I think I wrote that bio for Karen before I recently just started a new job. So I've kind of had a little bit of an untraditional, non-traditional career path, and I wanted to take a minute before I talk about my research to share a little bit of that with all of you, because I think as graduate students, you're in this position where you're at the very beginning of your career and you're doing some really cool stuff. But when I was in that place in my career, I felt like I didn't have a really good understanding of like the breadth of all the things that are out there that you can possibly do with your graduate degrees. So I'm going to take just a minute to share a little bit about that. So if it wasn't evident from the little intro or bio that Karen just read, I'm from Minnesota, and so that was also why I was very excited to talk to all of you today. I have a lot of friends that also graduated from the conservation sciences program for their graduate degrees as well. And if you don't already know how great Minnesota is, or if you're not with me on this, I might take a minute just to convince you. It's well, for one, as being in the Twin Cities, you guys might realize it's a really great place for culture and arts. It's the only state with public funding for the arts. From an ecosystem ecology perspective, it's also a really awesome place that's at the intersection of several different ecosystems. So we have the boundary waters with thousands of lakes in the boreal forest, and there's also the tallgrass prairie in the western part of the state, which is something that is super beautiful and enchanting, but I think it's often overlooked. And another thing a lot of people don't realize is that's one of the most endangered ecosystems in the world, with less than 1% of remnant tallgrass prairie remaining. So as you guys work on your degrees at the University of Minnesota, I hope you have the opportunity to explore and appreciate some of those places as well, particularly if they're outside of your study area. And as Karen also mentioned, Minnesota is the home of my alma mater, Carleton College, where I got a bachelor's in biology. And it ultimately wasn't until graduate school when I came out west to get my PhD at the University of Montana. And for my PhD, I studied population dynamics, population genetics, and trade-offs in management for a fish called the West Slope cutthroat trout, which is what the fish you see here on the screen. And this is a species that's actually a state fish in every state where it occurs, which is almost every Western state. And it's also a species of conservation concern. So I really enjoyed my PhD experience. It was like kind of at the intersection of both basic and applied research. And when I finished my PhD, I knew I didn't want to pursue a career in academia, but I didn't really have a sense of like all of the breadth of opportunities that were open to me. And so I was applying to jobs with kind of like nonprofit groups and some advocacy and, trying to look at some postdocs because those seemed like logical next steps. But ended up kind of in the midst of all of that, accepting what was supposed to be a short-term position with one of my committee members working on environmental DNA at the National Genomic Center for Wildlife and Fish Conservation, which is a lab that's housed within the Forest Service Rocky Mountain Research Station. So I worked as the eDNA program coordinator for actually a couple of years, and in that position, I did a lot of lab work. I did a little bit of field work, not as much as I would have liked, but and kind of at the core of this position was getting environmental DNA as a tool kind of up and running and accessible to managers in the National Forest System and beyond. And so if you're not familiar with eDNA, you will be after this talk today. So I'll get into that in just a few minutes. But in this position, I was, started out doing a lot of outreach, doing a lot of research to understand the limitations and advantages of this tool. But after about a year, it quickly kind of evolved into something that was much more like project management and kind of secretarial work with budgets and making sure people had equipment at the time that they needed. And it just wasn't really research. I didn't feel like I was using my PhD, using my education in a way that I really wanted to. So I worked with my supervisor who was my committee member and then became my supervisor to we kind of came up with a non-traditional postdoc that was soft-funded. So again, I was working to fundraise my salary and research dollars, but I was working as an aquatic research biologist and focusing my work on collaboration with tribes, tribal biologists and tribal natural research agencies. So as Karen mentioned, in this position, I was using genetic tools and information to inform management of freshwater fish, but with an emphasis on working with tribes. So why work with tribes? Why this emphasis? Some of you may have been involved in discussions or heard kind of reasons for why we should all be working with Native American tribes before. And if you haven't been, a part or listened to some of those discussions, I hope that what I share with you today is some inspiration to start looking into that. But in any case, why should we as natural resource researchers, educators, people working in this field be working with Native American tribes? There are a lot of reasons. The easiest justification is that there's simply legal obligations laid out in treaties for the Forest Service to collaborate with tribes, to consult with tribes when there's work being done on public lands that have tribal interests. But more than just these legal mandates, there's also, from a natural resource perspective, matters of efficiency. So what you're seeing on this map here is all Forest Service managed lands in green. And then in purple, there are Native American reservations. And you can see that particularly in the West, many of these areas are adjacent to federal public lands. And now I haven't even included on here national parks and Bureau of Land Management, BLM lands, which would increase the sort of areas where we have these political boundaries that are right next to each other. And from a fish and wildlife perspective, like animals don't care about these jurisdictional boundaries, right? And so we have these boundaries of public lands where fish and wildlife are moving across them freely. And we often have these tribal natural resource agencies and government natural resource agencies that have common objectives and goals for managing those resources. And so obviously there's a matter of efficiency. It just makes sense that we should be collaborating because we're going to be more effective in reaching those goals and managing these resources better with that collaboration. But beyond, again, sort of like this issue of efficiency and legality for why we have these obligations to work with tribes, tribes often have retained rights off reservations to manage fish and wildlife themselves, and also to exercise cultural heritage through take of fish and wildlife. So with this in mind, I wanted to kind of take a little bit of a detour and talk about some kind of current events going on. This picture is of a friend and colleague of mine, Lytle Denny, who is an enrolled member with the Shoshone-Bannock tribe, and he's also an anadromous fish biologist for the tribe. For those of you who aren't familiar, anadromous is a word that we use to describe fish with a life history wherein they're born in freshwater. They spawn and are born in freshwater, and then they go out to the ocean to get really big, and then they come back as adults to spawn again in that freshwater. And here Lytle is practicing subsistence harvest of Chinook salmon by spearfishing. And the tribes have a right off of their reservation to take Chinook salmon as part of their tribal heritage, part of their cultural heritage. And this year, the tribe was allotted a take of only 3 adult fish. So you can hardly call that subsistence. And I highlight this because this picture is from a Time magazine article titled "The Fight to Save the Salmon" that just came out this week on Monday. And so, Kieran, if you want to post that first link, Kieran will post a link to the article in the chat. And it's a really interesting article that discusses the decline of salmon in the Western U.S. and kind of the bipartisan struggle to gain support to protect these species with tribes as part of this fight. And it highlights some of the really important ways that these natural resources are connected to tribes and connected to this bigger picture of natural resource management. And then just yesterday, and I have to imagine that this is in response to the Times article, the Biden and Harris administration announced a kind of initiative to advance protection and conservation of salmon in the Columbia River Basin. And so, Kieran, that's the second link that I had. And I haven't read that full sort of outreach from the Biden-Harris administration, but kind of interesting to see some of this stuff happening, in real time, like this week as we're having these conversations. So, hopefully this kind of highlights to all of you some of the reasons that it's important to work with tribes. There are so many reasons. And on top of all of this, I also want to highlight that, above and beyond all of these sort of legal obligations and rights and mandates, tribes are the original stewards of our landscape in North America. And we have so much to learn from them. At times I find it's like just kind of both frustrating and ironic how we idolize naturalists that were really influential, like Aldo Leopold and John Muir, for their observations of the natural world, when tribes had made these observations and integrated them into their culture and well-being and their stories, and they basically knew all of this stuff for thousands of years before we feel like, oh, we discovered them or we made this observation. And so by not working with tribes, we're doing everyone a disservice, right? Like, we've reinvented the wheel for no reason. And we have so much to learn from them based on the many, many generations and thousands of years of observations and also living as part of the landscape. So we really should be working with them and incorporating them as we do our research and as we manage these natural resources. So if you're more interested in learning more about this, the best way to begin to engage is to educate yourself. And so with that, there's a cool tool that I think can be a good stepping stone or a good starting place for you getting interested in some of this. It's called the Forest Service Tribal Connections Map. And Kieran, that's the third link that I have there. It's an online map where you can click anywhere, and when you click on the map, it'll tell you whose homelands that location is in. That's a really great way. I encourage you to do that for your study area. Look at whose homelands you're working on, and then also just the places that you've lived. And you can, it's pretty detailed. I mean, the areas are kind of vast, where different tribes were kind of occupying land. But you can zoom in, and I was able to zoom in actually to the U of M St. Paul campus and click right on the campus to figure that out. So it's a really cool tool, and I hope you play around with that a little bit. So, all right, returning sort of to my career path history, as a tribal project coordinator, I was able to co-produce some really great research. A couple highlights were working with the Kalispel tribe of Indians to understand environmental DNA applications for assessing invasive species removal efforts and kind of lessons learned from that. And then if you came in early, you may have heard me talking with Nick a little bit about some northern pike work I've been doing. And so working in collaboration with the Confederated Colville tribes to understand both sources and mechanisms of invasive northern pike expansion in the Columbia River Basin. So I worked in this position for 7.5 years, and only just last month I started a new position as an aquatic research ecologist at the Aldo Leopold Wilderness Research Institute. part of the big reason that I made this shift is because this new position is a permanent federal position, so I'm not having to fundraise my salary anymore. And if you guys have questions about what that's like, I'm happy to talk about that as well. But there are a lot of things that I'm really excited about in this position. So this is an interagency group that's housed at the Forest Service, but performs research to inform stewardship of all designated wilderness areas. And we have multiple federal agencies that are sort of managing wilderness areas. So we're an interagency group. And one of the first things I want to highlight that is really exciting to me about this position is that our director, Jason Taylor, has a really big push for increasing and understanding ways in which underrepresented and underserved groups have been excluded from wilderness. And so as part of that, we're working on this one vacant position. We just hired somebody, a research social scientist, to specifically focus their research on these issues of diversity, equity, and inclusion in wilderness. And then another component of this sort of initiative or effort is that we have our writing informal commitments into our program of work to increase co-production of knowledge with groups that are underrepresented and underserved in a wilderness context. So that's all super awesome. The other thing that is both exciting and daunting to me about this work is that I'm the first aquatic ecologist that's joining this group in the 28-year history of the Institute. So I'm looking forward to bringing a new ecological perspective to the team, and then also continuing to elevate Native voices and knowledge through wilderness research. And one of the projects I'll be carrying over into this new position that has a wilderness relevance is the work that I've been doing on Pacific lamprey. So kind of bringing it back around to what I came here to talk with all of you about today. So some of you may be familiar with lamprey if you're spent much time in the Great Lakes region. Sea lamprey, which is a different species from Pacific lamprey, but sea lamprey are an invasive species in the Great Lakes region, but Pacific lamprey are native to the West Coast. Lamprey in general are an ancient lineage of jawless fish. They're completely cartilaginous, so they have no bones. And as I mentioned, they're native to the Pacific coast. Pacific lamprey in particular are native in North America to Alaska, Canada, Washington, Oregon, and also inland into Idaho. And this is basically overlapping with wherever we see salmon and steelhead. They also have a pretty cool life history that in some ways mirrors salmon and anadromous steelhead. The juveniles are born in a stream setting, but they burrow into the sediment and they live in the sediment, feeding off detritus and algae for up to 7 years. And so you can see here this juvenile lamprey at the top of the screen. It has no eyes because it doesn't need them. But when the timing is right, the fish will metamorph into subadults or macrophthalmia, and they'll develop eyes and also the ability to live in salt water, and they'll out-migrate to the ocean. At this phase, they're anadromous. So again, as I said, that means that they're born in freshwater and they go out to the ocean to get really big, and then they come back to spawn in the freshwater. But as adults, they are both anadromous and they're also parasitic. So they, Pacific lamprey will suck blood of other fish like salmon. I think there's also a benefit to this in that they get to hitch a free ride out to the ocean, or in some cases back to freshwater. After they get big, they'll return after 1 to 2 years in the ocean to spawn in freshwater. One interesting note is, unlike salmon, they don't home to their natal spawning grounds. So salmon will go back to the basin and often the exact stream that they were born in, and Pacific lamprey don't do this. So why Pacific lamprey would bother going far inland to Idaho is a little bit of a mystery, but there's just so much that we don't know about this fish. Historically, lamprey used to return in hundreds of thousands to millions of fish. So this is a picture from Willamette Falls, which is in Portland, Oregon, from a little over 100 years ago. You can see how many lamprey used to come back. It was just like in these droves. And so with these vast numbers of fish returning, and also on a pretty predictable cycle throughout the year, it's not surprising that Pacific lamprey were a really important source of food for Indigenous people of the Columbia River Basin. they're actually much more calorie dense than salmon. And so, while indigenous people of the Columbia River Basin also use salmon as a really important food source, lamprey were that much more rich and that much more, so they could get more out of them. So a really important food source, but also an important source of medicine and identity as well for the tribes in this area. So that kind of links in the interests of the tribes in helping to protect this species. But of course, part of the reason we don't know that much about Pacific lamprey is that kind of Western culture has not seen them as something that's valuable or enchanting or charismatic like the big Pacific salmon. And so we know so much about Pacific salmon and we know so little about lamprey. And one thing we do know is that lamprey numbers have declined dramatically. We don't see fish coming back, we don't see these lamprey coming back in these droves anymore. And there are a lot of reasons for this. Part of it has to do with habitat destruction in the freshwater habitat. So there's been historic mining and habitat degradation and spawning areas. But perhaps the most obvious and the biggest driver of their decline has been hydropower dams along our river systems in the west. And so again, here's that map of Washington, Oregon, and Idaho, and these are the major hydropower dams along both the Columbia River and the Snake River. And so, these dams have also been noted as a reason for decline of Pacific salmon. However, mitigation efforts to create fish passage for salmon don't work for lamprey because they swim very differently, right? Like they don't have the same body morphology and they move through the water in a very different way. And so some of the mitigation efforts to promote anadromous fish coming further inland and returning to these spawning grounds just hasn't worked for lamprey. So we're kind of not seeing any improvements in lamprey numbers from those mitigation efforts. So in 2003, Pacific lamprey in Oregon, Washington, and Idaho, and also California were petitioned for listing under the Endangered Species Act. And the Fish and Wildlife Service came back and determined that while lamprey had declined, there was not enough information to list them. Basically, there was insufficient information to form a recovery plan. And so without knowing even where Pacific lamprey still occur, they're not able to develop a recovery plan. And so they said, well, this isn't warranted. It may not be warranted right now. We just don't have any information. So, that kind of stalled efforts to recovery of lamprey and got researchers in the West kind of thinking and asking, what is it that we do know about lamprey? What do we know? What is the little information that we do have? So this is a map ranking subbasins where lamprey occur in the contiguous U.S. Historically, where they historically occurred in the contiguous U.S. For those of you who aren't kind of water hydrology people, subbasins are a unit of kind of a watershed. It's a particular level of a hydrologic code, and you can Wikipedia that and read all about it. But it's a nice way to kind of break up subbasins into units that we can focus on. So these are all the subbasins in the West where Pacific lamprey historically occurred and have been ranked based on what we know about their persistence. So the warmer the color, the more likely it is that Pacific lamprey have been extirpated from that basin. And it's not surprising that these inland basins are kind of where lamprey are the most imperiled, because again, those have far more barriers to fish movement for fish getting in there. Meanwhile, the coastal basins appear to be doing, okay. They're maybe imperiled at best, a couple of basins here and there where they're only considered vulnerable, but none where we're ranking them as apparently secure. And so the other thing I'd like to call attention to is these basins that are in black. And these basins just have no information to even rank them. So, this is a big kind of resonating theme here, is that lack of information is the primary challenge to protecting, conserving, restoring this species. So what is it that we need to know? Well, the first thing is just figuring out where they are. What basins are they present in? And then with the basins where they still persist, how widespread are they? What's their distribution across the basin? And once we have this information on the presence and distribution, it's going to be really useful to monitor that for changes in presence and distribution related to, for example, continued negative impacts on the species or conservation efforts to restore and expand the species range, and then as well as natural fluctuation in population sort of extent and numbers. But this is not a simple task. There are a lot of challenges because there's so much area that needs to be covered. A lot of this habitat is often very remote. There's a lot of wilderness areas in here that don't have roads, maybe just have trails or even no trails at all. And so it's difficult to access. And then also we have a number of freshwater lamprey species here in the West that at the juvenile life stage can be very difficult to distinguish morphologically from Pacific lamprey. And so even if you get your hands on juvenile lamprey in the field, it can be difficult to tell which species you're dealing with. So how do we overcome these challenges? Well, there is a magical tool called eDNA or environmental DNA. So some of you, again, may be familiar with this, but if you're not, eDNA or environmental DNA is simply DNA obtained from the environment rather than sampling the target organism itself. So what does the process of environmental DNA application look like? Well, we have a fish, in this case our lamprey, that's in a certain water body, and the presence of that fish in the water body, just by being there, it's sloughing off cells and DNA into the surrounding environment. And this can be from things like fish slime and epithelial cells and feces and gametes during spawning. So they're exuding their DNA into the environment. And then what we do is we just capture that DNA by filtering the water. And the DNA is captured on a filter where we can bring it back to the lab and test that DNA or analyze that DNA for the presence of various species. Now, currently the best and most accurate method for testing these samples for the presence of a particular species is quantitative PCR. And I mention this because for those of you who aren't sort of genetic nerds out there, Qpcr has become a buzzword in the context of the pandemic. So Qpcr, or quantitative PCR, as many of you probably are familiar, is what's being heralded as the most accurate test for COVID. And it's exactly the same technology, exactly the same test that we're using to analyze our eDNA samples. So how does this work? The same test, and in this process, what we do is we combine our samples, so either a nasal swab or the DNA extracted off of the filter that we sampled that water. And we combine it with a molecule that is specific to the unique DNA of our target. So our target may be Pacific lamprey, so we're looking for a DNA sequence of Pacific lamprey, or we're looking for the DNA sequence of the coronavirus. This assay, this molecule, or what we call an assay, contains a fluorescent component so that when the assay comes into contact with our target DNA, we get the emission of light. And that's basically what we're looking for in the lab. Is we run this sample, and when we see the emission of light, we can say, aha, that person does have COVID, or aha, Pacific lamprey were present in that sample. But if a sample is negative, meaning we didn't have the DNA of our target, whether that was coronavirus or lamprey or some other fish, we just see no light. So that's kind of how that process works, but knowing whether an individual sample is positive or negative really means nothing until we put it into context. So for example, with COVID samples, we're trying to track infection perhaps over time or over space. And similarly with eDNA samples, we're trying to figure out where are lamprey on the landscape, right? So that's it. That's the last step of eDNA in this whole process. And what makes this tool really useful is that it is highly specific. So it is distinguishing between DNA of different lamprey species. And it doesn't matter if we have freshwater lamprey also present, it's going to pull out that Pacific lamprey DNA. It's highly sensitive, so it can often pick up the DNA of organisms that are present at very low densities when it may be otherwise really difficult to capture them with other methods. And then finally, it's super efficient. So it only takes about 10 to 15 minutes to collect an eDNA sample. The most time you spend is just getting to the site. Other traditional sampling methods where we need to actually have a fish in hand require many more people and are much more time intensive. So with this, we can sample broad landscapes really efficiently. So with all of this in mind, it seems like eDNA is kind of our golden ticket to starting to understand lamprey presence and distributions on the landscape. And so we've been, my co-PIs and I have been working on a project that we're calling the eDNA Basin-Wide Lamprey Inventory and Monitoring Project, or eBLIM. So without being able to see your faces, hopefully this joke lands a little bit, but in any case, we found that it's easy to have good acronyms for projects that you're working on. So maybe you, I can encourage you guys to come up with that for your own theses and dissertation studies. But the eBLIM project, so what's our study design? Remember, we're trying to figure out where are lamprey, and we're not just going to run around and collect eDNA samples everywhere, right? We want to have some guidance on where we're collecting samples. So the first thing we thought is, well, let's figure out where lamprey like to live. What are their habitat preferences? My co-PI, Dan Isaac, developed a model by looking at publicly available data on Pacific lamprey in the Western US and kind of summarized what he found about the habitat where they were present. The biggest drivers of where we're finding lamprey have to do with stream flow, so how much water, stream slope, how steep is the stream, and then the water temperature. And there's a little bit of an interaction here. So we don't find lamprey in very cold waters, no matter how steep the stream is. But otherwise, lamprey tend to like waters. There's a higher probability of lamprey occurrence when the water is a little bit warmer. And then we generally don't find them in very steep areas. So headwaters, those bigger river systems. We can take this information and then apply it to a map to see, okay, well, where are the stream and river segments that have these habitat qualities? And that's what you're seeing here is our map of streams and rivers where our probability of lamprey occurrence is greater than 0. So that's great. We've pared down kind of where we would like to sample a little bit, but this is still quite a big area for us to just go out and collect samples. We can't just run around and do it everywhere. So how did we narrow this down further? Well, we took a focus on subbasins, again, those fourth code hydrologic units in Idaho, Oregon, and Washington. This is in part because this is where there's the most inland habitat. Lamprey appear to be the most imperiled, and then also where the funding came from. So we had some funding from the Forest Service and the BLM to specifically focus on sensitive species like lamprey in this region. So we overlay our map of suitable lamprey habitat, and we thought, okay, we want to sample this coarsely. So within these subbasins, we're going to sample the main stem every 15 kilometers, so long as that location has a probability of lamprey occurrence greater than 0. And then we're going to sample tributaries to those main stems. If, again, the lamprey probability is greater than 0, we'll collect a sample near the confluence of that tributary. Or if that stream is known to support Chinook salmon. And this addition of Chinook salmon was intended to kind of broaden our scope a little bit. We wanted to be sure that we could account for the places where our model is wrong, because no models are perfect, right? We need to expand our sideboards a little bit to kind of figure out where our limitations and errors are in the model and correct that. All right, so now still a task of getting out there and sampling, and we were able to kind of narrow this down, but it's still quite a big task. And so in order to do this, we appealed to partners and interested groups at a very large number of organizations. So we had many tribal partners, we had local watershed councils in Oregon that helped us out, many individuals from local, state, and federal government agencies, and then as well as some nonprofits as well, getting out there and helping to collect samples. We started sampling in 2018, and to date, we have collected almost 1,000 samples for the eBLIM project. This is a map showing all of those sampling locations for the eBLIM project, overlaid with those nature reserve rankings. So you can see kind of where we're sampling in relation to what we know or maybe don't know about lamprey persistence. Now, what did the detections look like? Here is a map of the detections. The red triangles are positive detections of lamprey DNA, and the gray circles were non-detect. So this is great. There's a lot of information here. Let's look a little bit closer in detail in inland Idaho. This is as far inland as lamprey occur across their range. This is what we found in Idaho. The blue areas are, again, the segments that have greater than 0 probability based on our model of lamprey occurrence. And what we did in Idaho was a little bit two-step. We had these coarse sampling, this coarse sampling effort, 15 kilometers in the main stem, and then at the base of tributaries with a non-zero probability of lamprey. We started that in 2018, and then in 2019, we wanted to go back and really understand, okay, how far inland are lamprey occurring right now? And so we went back and sampled tributaries with positive lamprey detections at 1 kilometer intervals to get a much finer resolution. And basically what we found is the only tributaries that, well, so generally we found there was very limited tributary occupancy, and the only tributaries where we had positive detections very far upstream were areas where the Nez Perce tribe had been performing reintroduction efforts for the species. So that's really great. We're able to help validate that those reintroduction efforts are, it looks like they're working, and then also kind of identify quite a lot of vacant habitat for future reintroduction efforts. We also found that wanted to document the upstream extent in main stems, and that was in the Salmon River near the town of Carmen, Idaho. For reference, Missoula is right here, kind of at the edge of this inset map. And so if I drive about 2 hours south, I theoretically could go and see a lamprey in the wild, a Pacific lamprey in the wild at its furthest inland location. So that's what's going on in the inland areas. What about the coast? Let's take a look at 2 of our coastal basins, the Siletz, Yakima, and the LC. This is coastal Oregon. So unlike inland Idaho, which is a huge, really long river systems that eventually drain into the Columbia River, which eventually drains out to the coast, these are very small river basins because they're just flowing off the Cascade Mountains here in Western Oregon. So we collected our e-blimp samples, and one of the great things about having a project like this where you collaborate with partners is partners are often able to leverage funding and leverage opportunities to collect samples of their own. So through that, we have our e-blimp samples in blue, and then more, again, another opportunity for fine scale sampling based on the needs of the Mid-Coast Watershed Council and the Bureau of Land Management. And this is what we found. It was really great to have that leveraged opportunity for more sampling because we were able to get upstream of lamprey in some of these coastal basins as well. And one thing that was surprising to us here was how many positive detections we had. And that's not because we knew that lamprey were kind of doing better on these coastal areas, but what was surprising to us are the number of locations where our model said, yeah, the probability of lamprey is greater than 0, but it's not super high. So this is 166 samples in total across these 2 basins. And of those, all of these samples in green are areas where our model said, yeah, the probability of lamprey is greater than 0, but the probability is still less than 50%. So why is that? Our model is under predicting occurrence. Well, part of it might be because the publicly available data that my co-PI Dan was able to get to build this model is primarily from inland Idaho. So a few things going on there. The habitat in inland Idaho is really different in many ways. It might not just be about stream temperature and slope. There could be other factors that are different here on the coast that we're not accounting for with the information that we fed into the model. The other thing is that there are simply more lamprey on the coast because there are fewer barriers to upstream movement. And so where we have higher densities of lamprey, they're going to be occupying more habitat. There are going to be more fish that are potentially occupying habitat that's not, perfect, like 100% ideal, but like, hey, it's still pretty good. So where we have higher densities of fish, we're just going to see more occupancy. And so we think that could also be what's going on here. So this project is not yet complete, but I can give you some insights into some summaries and observations of Pacific lamprey. So to date, we've collected, and this doesn't include samples from this past summer, but as of 2018 through 2021, we collected almost 850 e-blimp samples, and then working with partners, we're able to leverage even more than that. So have quite a lot of information across many areas, and through those collaborations, we're able to sample quite a broad range. We noted limited tributary occupancy in Idaho, except where reintroduction efforts on behalf of the Nez Perce tribe have occurred. And we also noticed that our model is underestimating lamprey occupancy in coastal habitat. So another cool thing about this is all of the data is being made publicly available on an online map called the eDNA Atlas. And so Kieran, that's the last link I had to post in the chat. If you want to go play around with that, it's pretty cool. All of the eDNA samples that have come through the National Genomics Center are being posted on this map. And so when you go to the map, it's an interactive map, so you'll have to kind of expand this section on the right, and you can click on various species and see where samples have been analyzed for that particular species and whether or not they were positive or negative detections for that species. So making this data publicly available is going to make it really important to just get the information out there, right? We're not waiting for a publication to come to get the data for a publication, because it's just more meaningful from a management perspective to just get it out there so everyone can use it. And then this year, I'm really excited about the samples that we have trickling in. They're mostly coming in the lab, but they have not all been analyzed yet. In pink, you'll see the samples that were collected this year, the proposed sampling locations. So we're starting to inform many river basins here, especially around the Puget Sound and in northern Washington, northwestern Washington, where we have no information, no idea about Pacific lamprey. Yeah, getting some of those gaps filled in is going to be really exciting. And again, the data from this study, from this work, is already being used by tribal, state, and federal biologists to help protect lamprey. And ultimately, what we want to do is keep them off the endangered species list. So they were petitioned for listing in part to get, being on the endangered species list is like being in the ICU. You get resources and treatments that can help revive a species and restore a species. But ultimately, if we can get enough information out there, it's better if we can keep them out of the ICU, right? Keep them off the list. So we're hoping that this can be a really big stepping stone in that effort. So with that, that's the end of my presentation, and I'm happy to take questions. All of you, if you have questions about anything I've presented today, please feel free to contact me. I love engaging with students. I also just wanted to show a quick picture of lamprey. I was able to have the opportunity to go help the Nez Perce tribe sort adults before a reintroduction effort, and so it was taking some genetic samples, helping to collect genetic samples and then sort the lamprey. And the suction cut power of their mouths is really incredible. And so, I think everybody here was like kind of engaging with the fish to see what it was like to have them suck on you or hold on. They use that suction power not only for feeding, and to be clear, this fish did not bite me. It wasn't predating or parasitizing me in this photo, but they also use that to climb rocks. So they'll like suction onto a rock, suction up, and then kind of propel themselves forward. So a way that they are able to get over some barriers. Cool. So yeah, thanks for taking the time to listen to me today, and I'm happy to take any questions.
Awesome. Yeah, thank you, Dr. Carim. That was really, really cool. I guess I'll just say, so we do have a couple questions in the chat right now. So I'll get into those, but if anybody does want to unmute and ask a Zoom face to Zoom face question, please feel free. It's always nice. So I'll start with, one of the first questions here. Lucas asks, what is the average cost per sample for eDNA?
Yeah, that's a really good question. It kind of depends on the scale of the project. I think that the lab, if you have a large scale project, it's like, it can be a little more flexible, I think, with like, what are the deliverables? What do you need from the lab kind of thing? Is it just straight up sample analysis, or do you need a report? Are you looking for, collaboration on kind of what scale? But I think at a really basic level, it's about $85 for a sample. And that includes like the materials that you need to collect the sample, because eDNA is really susceptible to contamination. And so the lab will provide that because they know that it's clean and like free of contaminants. And then that includes analysis for a single species. One thing that's super cool about eDNA is it doesn't have, it has DNA of everything that's in that proximity of that sample location. And so you can analyze the sample over and over again for multiple species. And once the sample is collected, it's much cheaper to do that because you have you have the DNA already extracted, so it's a cheaper process.
Yeah, that's thank you for that information. I mean, I guess building off that a little bit, how you'd imagine that there's just a massive amount of cost savings when you compare it to actually going out and doing, like sampling, getting a team out there and doing fishing or electrofishing or something like that. So do you have any estimates on how that compares necessarily and the utility of that?
Yeah, so we haven't calculated that number in our lab internally, but there are some papers out there that have done sort of similar estimates. I haven't seen one. I've seen anything from it's 4 times cheaper to 2.5 to 4 times cheaper. There's a study looking at one of the really early studies on eDNA, looking at invasive bullfrogs in France. They estimated that it was 2.5 times faster and I think 4 times cheaper. But the other thing is that it didn't require expert knowledge of species identification in the field, right? So there's some added advantages too, where it's not even just time savings, but sort of preventing errors in certain instances if you have people that are unfamiliar with the species. And I've definitely had that too, where people have said, oh my gosh, there was definitely brown trout in this stream. And it's oh, it's not really brown trout habitat. And then they collect an eDNA sample and it's oh, it was brook trout. And the person that was in the field just wasn't as familiar with what brook trout looked like. And there was a misidentification. So sort of forensics, the DNA doesn't lie.
Very cool. And then another question in the chat, does any of your work, and this is from Lauren, does any of your work get at eDNA copy number relative to fish abundance?
Yeah, so yes and no. I think that the question of kind of exact fish abundance or biomass is something that can fluctuate so much with a particular system. DNA degrades over time. It degrades with UV light. It degrades with temperature as enzymes in the water kind of eat it up and destroy it. But it can also be retained in the environment in an anoxic, environment and bind to sediment and things like that. So it's this very dynamic process. So coming up with a model to estimate exact abundance is, it's something that I think we're just not going to be able to do with this tool. Right? But eDNA can be really useful for looking at a lot of fish or a little fish and looking for changes over time. So we've definitely used it to look for, say, hotspots. If you have just a little bit of DNA, it's pretty safe to say you don't have, a population level abundance in close proximity to where you sampled. But if you have a lot of DNA, then yeah, you probably have a lot of fish there, right? And then as you collect those samples and again, put them in context. So collecting samples at regular intervals throughout space or at one location at regular intervals throughout time, you can get a sense of okay, yes, there are much more fish here than there, or there are much more fish at this point rather than that point. So it's a loose association. There's definitely an association. You can definitely get information, but for things like abundance estimates, you still have to get in there and actually sample fish.
Yeah, that makes a lot of sense. And so I guess, Roger, did you have a question that you'd like to ask?
Yeah, I have a couple questions. First, I want to say go Carleton. I'm an alum myself, graduated in 2019, and I'm also working with tribes on a couple projects related to chronic wasting disease in the region.
Interesting.
My first question is, how are you and your agency building and maintaining collaborations with these tribes? And then my second question, I guess it's a sort of multiple questions. Given that you're working with culturally relevant species and maybe even areas, are there any complications or things to be aware of or needs of the tribes or maybe even different approaches that you have to take? Could you just generally speak on that?
Yeah, so, and I'll definitely interject if I'm kind of not answering your question or yeah, going down the wrong road. But so first, what is the Forest Service or the different agencies doing to engage with tribes? Yeah, this is it's difficult. It's a difficult question because the federal government is so huge, right? And we're often at the sort of whim of what gets funded and what gets prioritized at a congressional or presidential level. But the Forest Service does have an Office of Traditions, which is focused on engaging with tribes on public lands and meeting those treaty rights. But honestly, a lot of this stuff gets overlooked, right? Because it feels good, but it's, in my experience, like it's not always the priority. And I think we all see that. And so it's really difficult. And the other thing that a lot of people don't realize is that working with tribes takes time, and there are cultural differences that are often not appreciated, right? And it's, you know, a lot of points in my career working with tribes, it's like, okay, well, like I recognize that the time I'm spending building this relationship, which is really important, means that I'm not getting publications out in this other realm, right? And like, and it's frustrating because it's like you don't get credit for that, but it comes up oftentimes to like the will and interest of individuals in many cases. But I'm hoping that, you know, the Biden and Harris administration has put a lot of emphasis and initiative on helping to engage with tribes and do a better job of that. And then, you know, for me, in my new position working at the Aldo Leopold Wilderness Research Institute, my supervisor has specifically outlined like diversity, equity, inclusion, and specifically, you know, with tribes, because of their connection to the land, is something that he is working really hard to prioritize. And that's something that is so important to me, like honestly, on a personal level. It's really great to see that happening. So I don't know if that's kind of an unsatisfying. There are a lot of individual programs, but the government is just so big. And then your second question was kind of related to what are kind of methods.
Yeah, sort of like how does your research change when you work with them, because you might be working with a culturally relevant species or area, or like, are there any complications or things that you should be aware of, or others should be aware of, or are there different needs that need to be recognized?
Yeah, that's a great question. So there are different needs that need to be recognized, and there's certainly like working with culturally relevant species, and also broadly working with tribes, things that you may not consider and may not be taught to you, right? Like in school necessarily. And so one of the challenges I've faced is engaging with tribes from a the perspective of using genetic tools and information. There are some tribes that have been horribly discriminated against and have had their genetic information, like human genetic information, used in ways that is extremely offensive and discriminatory and inappropriate, right? And because of that, and this has happened within our lifetime, right? Like this was in the 90s. And so this is like when many of us were kids, and to think that I'm trying to engage with tribes and build partnerships with tribes on the basis of like saying, hey, let's use genetic information. And they're like, no, you know, like I have no interest in that. So, you know, that's one example of where it's just like, okay, well, I'm, you know, what I can do is tell you how I can help, but I can't force anybody to work with me, right? But when you're working with tribes, recognizing having this sort of cultural competency, cultural sensitivity, and recognizing that they're coming from a different perspective is really important. With certain species, like one of the great things about environmental DNA is with certain species, it's like, it's considered sort of like bad luck to actually handle the species, or you don't want to actually touch it, right? And so eDNA is a great way. It's a non-invasive tool that allows us to do that with other tribes. You know, the bigger thing, and maybe less specifically to culturally significant species, is just how you engage with tribes, spending a lot of time listening, talk less, listen more, right? And just spending time in general, taking those opportunities to go spend time with people in their homelands and learn. Tribal partners have a very different way of looking at the world, looking at the ecosystem, because that's like indigenous knowledge, right? And I'm not native, I don't have that, I wasn't passed on that knowledge. And so being able to spend time with them, engaging in their knowledge process, their ways of knowing, is really important. And also recognizing that the point of combining and co-producing information is not to take indigenous knowledge and insert it into Western knowledge, right? Like that's not co-production, that's not what this is about. This is about two systems of learning and how we can learn from one another. And so being conscientious of that, being conscientious of the time it takes, understanding that working with tribes means like everything's not going to happen on your one-year funding cycle, right? Like being patient and taking the time and really just having faith and knowing like, okay, yeah, you know, maybe I didn't get publications out this year, or maybe I was delayed on a publication, but what I'm doing is the right thing, and having faith in that too.
Thank you.
Yeah.
Yeah, great questions, Roger. And yeah, thank you, Kelly. Those are, it's a lot of really good information for us for sure. If anybody else has any more questions, please feel free to unmute or put them in the chat. I have a couple that I'd like to ask if cool. So, you showed some interesting kind of maps, of distribution and kind of mapping out the probability of, the Pacific lamprey being in certain areas. Do you have any comparisons to like how damming, actually influences the probabilities, like a before and after shot of that, or like how much more potential, rivers or streams could be in that if we were to actually remove some of the dams or that are put in place?
Yeah, that's a really good question. No, we don't. And so like the model was only looking at like the habitat suitability. It didn't incorporate barriers to movement. It was just looking at habitat suitability based on like those like stream characteristics at a particular location. But now that we're doing all the sampling broadly, I think we'll be able to, it's possible that we'll be able to incorporate that, right? And say, okay, here are samples above and below known barriers, like known dams. But in general, I imagine like there's a lot of talk right now about taking out some major dams on the Columbia River Basin. That's going to help all anadromous fish. It's going to help all of them. And so it's, the article, the Times Magazine article that I posted is really interesting in that respect, talking about like the duality of these issues and like the bipartisan, the politics, and it's green energy. So like hydropower energy is like good, but it's bad at the same time. It hits on a lot of those topics.
Very cool. So we are right at 1 o'clock here. And I think we'll, if anybody does want to follow up with Dr. Carim, please feel free to do so. And, I think you can provide a lot of really pertinent information for sure, especially in the Pacific Northwest and some of these species. I guess we will have a conservation sciences presentation again next week. So please feel free to join us. And I just want to thank you again, Dr. Carim. We really appreciate you joining us today and look forward to reading up on your research in the future.
Oh, great.
So thank you.
Yeah. And please email me, everyone, if you have any questions. I'm always happy to hear from folks. So and have a great weekend.
Yeah, you as well. Enjoy.